Bismillahirrahmanirrahim. Assalamu alaikum. In this video, I'm going to talk about the types of castes. We have these 4 to 5 types of castes, and in the previous video, I have talked about the basic concept that what are castes and how they get formed, the theory on their formation. Now, in this video, we are going to elaborate each type of caste. So let's begin. The first type of caste we are having is Hayline caste. Basically, it forms in the absence of cells in the tubular lumen, and it consists of TAM, horsefall, mucoprotein only. So, in this caste, there is no cell, just TAM, horsefall, mucoprotein that itself precipitates and that like forms this caste. It is very difficult to see in the wet preparations of urine and must be distinguished from the mucus strands. As you can see this image over here, that this is a wet preparation, and I hope you guys know what a wet preparation is. Basically, it's a type of a preparation in which you put your liquid sample onto the slide and you view it under microscope being wet. Like, we do not dry it, fix it like we do in some other slide preparation methods, where we basically pour our liquid sample on the slide, fix it, stain it, dry it, and then view it. No. In wet preparations, we have that liquid sample on our slide and we directly view it under microscope. And that's why in the wet preparation of the urine samples, hairline casts are very difficult to view. Like you can see, they are very light in color. They have parallel sides and have clear margins and blunted ends. This is an important thing to remember, that these casts have parallel sides, like they are parallel, and not only that, their ends are blunt. Like this is a blunt end. So similar over here, it has blunt ends. Where mucus strands are more variable in size and have irregular margins. So, they have basically distinguished or like given a difference point, that when we talk about hairline cast, they have clear margins, parallel sides, and blunted ends. But mucus strands are variable in size and have irregular margins. So basically, you should they often are being or like wet techs basically, or wet students basically confuse these two when they observe it under slide, but you should remember this difference and differentiate these two. Greater number of hairline casts may be seen in association with protein urea. So in the cases of protein urea, because horsefall is a mucoprotein, it's a protein. So in case of increased protein in urine, you can see hairline cast. And there are two types of cases where we can have protein urea. Either it can be pre-renal or it can be renal. In case of pre-renal, there may be overflow of protein urea as a result of myeloma, or in case that you are feeding an animal high protein diet, like in like there are several reasons uh, of pre-renal increased pre-renal protein urea. One of them is this. Similarly, as far as renal concerns or renal indications of protein urea are concerned, there this might be glomerular disease. So whenever in your exams you hear glomerular disease, this hairline cast must come in your mind. But you should remember that not all cases of protein urea are going to have hairline casts. So this was all about hairline cast. Next we have is cellular cast. In cellular cast, cells are incorporated in TAM horsefall protein, forming cellular cast. So previously we saw that when it was hairline cast, it was just this protein and it was forming hairline cast. But when we are talking about cellular cast, in the same TAM horsefall protein, the cells are being incorporated. Just like you can see in this picture, these all structures are cells. These cells are basically sloughed off from the tubules of the kidneys. But there are two ways, like why these are like sloughed off, because of any degeneration or necrosis that can happen in these tubular epithelial cells. And the degeneration and necrosis can occur because of either these three reasons. Reason number one is there is any ischemia or infarction, that the blood supply to these cells is being stopped or seized, and because of that there is necrosis or degeneration. Or there is decreased renal perfusion, or there is oliguria secondary to dehydration, like animal is drinking less water, because of that the perfusion to the kidneys is less. But this case is like not that like tricky, because if you will rehydrate your animal, your normal urine volume in the animal will be restored, and that will flush these casts out. So this can be dealt easily. And number third thing is nephrotoxicity, like you have fed animal any of the you can say drug, like we have aminoglycosides that cause nephrotoxicity. They will cause the degeneration and necrosis of tubular epithelial cells. These cells will start to slough off and cause the cellular cast. These casts basically indicate acute tubular injury, but it does not indicate the extent and reversibility of the injury. So they do indicate that there is any ongoing injury, but what's the extent? You are not gonna get the idea from these casts. These are one types of cells that are epithelial cells that are present in these casts. There are another types of cell that are leukocytes that can be present in the cellular cast. And what are the indications that will cause the leukocytes to embed in these casts? Is number one, tubulointerstitial inflammation. Just like in the case of pyelonephritis, the leukocytes will increase in the urine and form cast. There is also at this point really possible to distinguish between epithelial and leukocyte cast, as nuclear detail is obscured by degenerated state of these cells. So what they are saying is that yes, we have two types of these cells in the cellular cast, either it can be epithelial or leukocytes, but under microscope it's difficult to see that either it's a cellular cast made from epithelial cells or it's a cellular cast made from leukocyte cells, because the leukocytes, although they have very prominent nucleuses, and even in the CBC under slide of a blood sample, we differentiate WBCs based on their nucleus. But here the nucleus detail is being obscured because the cells are in a degenerated type of a state, so it's difficult to distinguish. But you should know as a good veterinarian that these two cells types can appear in cellular cast. Next we have is red blood cell cast or hemoglobin cast. Basically these are, you can say, subtypes of cellular cast, because red blood cell is also a cell, because they have a different color variation, so we are mentioning it in a different slide. RBC casts are due to intrarenal hemorrhage, where RBC get caught up in TAM Horsfall matrix, forming an RBC, forming a RBC cast. So RBC basically start to degenerate in this cast, and when the RBCs will degenerate, it will change into a hemoglobin cast. So if the RBCs will degenerate, they will form a hemoglobin cast. These casts are rarely seen in animal or human patients, so you should also know that they will appear in rare cases. On a wet preparation, these casts basically mimic granular cast. Just like you can see these images, like it has almost similar appearance like granular cast, because we will see granular cast in the next slide, so you might not be able to differentiate, but in the next slide you will see that this cast looks like granular cast, but there are few differences from which we can have the difference between a hemoglobin or a red blood cell cast and a granular cast. And what are these differences? That the RBC casts have characteristic yellow red appearance in because of hemoglobin, and whereas granular casts are always colorless until they take on bilirubin stain in animals, if the animal is having bilirubinuria. So the color difference will help you differentiate between a red blood cell cast and a granular cast. But if the animal is also having the like a pathology of bilirubinuria, increased bilirubin in urine, it will going to be a bit tricky for you, but still you can differentiate. On a right stain, the hemoglobin within cast is muddy purple. It's not a, let me tell you that this point is not that important, but still I have mentioned over here, like if you are not a student who is going to give an exam, rather you are working in a lab, and you should be aware of this thing, that using right stain, the hemoglobin cast is a muddy purple rather than being red in color. And not surprisingly, if you stain it with Prussian blue, it is not going to give you a positive result, because Prussian blue normally stains hemosiderin, not like it will not give you a good positive results, stain positive result. So that was this point. Next thing is, what does these red blood cells cast indicate? Basically, they indicate any intrarenal hemorrhage, that there is any bleeding happening, any glomerulonephritis conditions, acute tubular necrosis, renal ischemia, and there are many other conditions. Also, hemoglobin casts, particularly their Waxy variants. What are Waxy variants? You will see in the Waxy cast slide later on. But hemoglobin casts are also seen with hemoglobin uric nephrosis or acute tubular injury from intravascular hemolytic anemia. This is just like, you should know that these all are the conditions in which you can have RBC or hemoglobin cast. I hope you guys know or have a bit of a knowledge what hemoglobin uric nephrosis is. It is basically a condition in which there is like bit inflammation of your kidneys, especially the filtration side, the Bowman's capsule, and the pores will get increased, so that they will start to filter the hemoglobin. To normally, if hemoglobin is a large molecule, it does not get filtered, but in the case of nephrosis or nephritis, the hemoglobin will also start to filter, and that's why they will appear in urine and form a hemoglobin cast. These were all the indications of hemoglobin cast. Next, you should see this image. Like these two are the images of hemoglobin cast. In these cars, this is basically a urine sample in which you are seeing these cars. You can see there are a lot of these cells. Basically, these are RBCs. These are also all RBC cells, because it's a RBC cast. So definitely, not only in the form of cast, you are also going to be see RBCs dispersed in the urine sample. Why? Because there is a lot of blood coming in this urine. And one thing I wanted to tell you guys about this RBC, in the wet preparation of urine sample, that you will see that RBC is never gonna take up the stain. It will always gonna be like this type of transparent type of cells, small cells, and you will recognize them as it is. That even in this slide, you can see these all are RBC, but they haven't taken up any stain. If there is any large cell, basically it's not a large cell. It might be macrocytic RBC, but these all are RBCs because they are not having the stain. Okay, next we have is granular cast. Granular casts are the casts that have textured appearance, which ranges from fine to coarse in character. Um, Basically, you can see this cast. This is a granular cast, and if we go back, now you can see this RBC cast and then this granular cast, and you can see there is a marked resemblance in both, and you can only differentiate by these casts being colorless, or the trickiness will appear if this cast, granular cast, or the urine sample is going to have increased bilirubinuria, because increased bilirubin in urine is going to give these casts still brownish color, so it will get difficult to distinguish, but still they look similar. You can see, we have also discussed this point earlier in the previous lecture, that granular casts are formed as cellular casts degenerate. You should remember this picture from previous slides, previous lecture, that cellular casts are formed first, they degenerate into coarse granular, which degenerate into fine granular, and which degenerate into waxy cast. So this is a sequence. Also, fine granular casts in low numbers, they do not indicate any pathology. They may also occur after strenuous exercise. But if you see coarse granular cast, they do indicate renal pathology, and you should get alert that it's a red flag if it gets appeared in your animal's urine. Next we have is fatty casts. Fatty casts are the casts that have lipid droplets within the protein matrix. It's very evident. Fatty casts are like granular casts, represent tubular degeneration. So basically these two both represent tubular degeneration. The background matrix of fatty casts are haline or granular in nature. So background is going to be either haline or granular in nature, but only what's the difference between these two and fatty casts is that if in these two casts fat droplets get embedded, it will convert to fatty cast. Like you can see these 2 points, that if hyaline casts have fatty droplets embed in the matrix, then it will become a fatty cast. But if you have a haline cast that is only surrounded by fatty droplets, but these fatty droplets are not embedded, like they are present around this cast but not embedded in this cast, then it is not a fatty cast, it's rather a haline cast. You can see in this picture too, all these transparent type of molecules like circle spheres are basically fat droplets. Um, so the fat basically appears like this bright lightish color and in spherical forms under microscope. Those who have seen themselves under microscope are well aware. Then lipid in urine. Now it's like kind of an indication like why fatty cast formed, or like how fat gets into the urine. Uh, yeah, the lipid can be present in urine as free lipid due to lipiduria. Like there is increased lipid excretion in the urine, and the fat or lipid is present as free, and it gets embedded in haline or granular cast, forming a waxy cast. But the second indication is the lipid can appear in urine due to degeneration of sloughed renal tubular or or transitional epithelial cells. So basically, epithelial cells are being sloughed, and because of their sloughing, the fat or lipid droplets from these cells start to appear in the urine, because the cell membranes contain lot of lipids, and this the sloughing of these cells can be physiological or pathological, like in the case of renal tubular injury, because physiological shedding of the cells also occur in the body, because every organ has a turnover rate, and after that it sheds its own old cells and renews and forms new cells. So it can be physiological or pathological. Next point is about cats. You should remember, whenever fatty cast comes in your mind, this point should come in your mind, that it is most common in cats, because as their renal tubular epithelium can normally contain small to moderate amount of fat. So because of that, in cats, most common cast is fatty cast. So that was about fatty cast, and here we are on to our last cast, that is waxy cast. It is final stage of cast degeneration. It originates from cellular or granular cast, which will either start to degenerate and form waxy cast. Compared to haline cast, they are readily observable and have smooth appearance, no internal texture, and are more refractile than surrounding. Basically, what does refractile means, that these casts are having transparent or like shiny or glassy appearances, just like you can see it in this figure, that these are transparent glassy appearance they have, and they have squared off ends and look like as if brittle and easily broken, and have smooth parallel sides. Like you can see, they have smooth parallel sides, and they have squared off ends. Uh, and if we compare waxy cast with haline cast, we remember the haline cast used to have similar parallel sides, but they were having blunt ends. Like they were having these round blunt ends. But waxy cast are not having blunt ends. Rather, they are having smooth squared ends. So that's a difference point between them. Waxy casts do indicate a tubular injury of a more chronic nature than a granular or cellular cast. In the previous lecture on the cast, I have talked about that these three casts, granular, cellular, or waxy cast, basically indicate chronic injury, or they indicate tubular injury. But Waxy casts specifically indicate a more severe chronic injury and likely associated with low urine flow and always of pathologic significance. So they are always going to be of pathologic significance. And this picture you are seeing over here is basically has not only waxy cast, but it also shows granular cast in it, like this one. And this is a urine sample of a dog, and it indicates acute and chronic renal tubular injury. So this was all about all types of cast. This is an image that I came across while searching pictures or clip arts on the Google while making these slides, and I loved this image. I loved this picture, and I thought to add it in this slide because it sums up all what I have until now elaborated to you guys. Under microscope, if you are going to see mucus strands, you are going to see it like this. I have told you they are going to be irregular in shape. Then Hayline cast, parallel sides, blunt ends. Then Waxy cast, parallel sides, squared off ends. Granular cast, two types, fine or coarse. You can see the difference. Then there might be bacterial cast if there is a lot of bacteria entrapped in TAM, horseful protein. Then hemoglobin cast, you are going to see all reddish tint, but you are not going to see any cells. But if it is a RBC cast, you are going to see RBCs, and you can see I have told you RBCs do not pick the stain. They are going to have yellowish brownish tint, but still they are not going to pick the exact stain, so even they haven't colored it. Then we have leukocyte cast, that is a cellular cast, lot of cells embedded. Then you can have renal epithelial cast, you will going to have large epithelial cells in the cast. And then we have a fatty cast, where we have fatty droplets with bright whitish translucent type of image. So I love this image, and I hope you guys have like well understood all what I have elaborated up till now. The video has got a bit long, but it was worth it. And this image, I loved it, I loved it so much. So guys, that was all about cast, types of cast. I hope you guys liked this video, guys. Content making takes a lot of effort, so do like, subscribe, and share this video. Thank you so much, thank you so much for watching Vet Fraternity.